मतलब कोई कमी मिल रहा होता है, ये दे भी नहीं रहा। फिर इनके पास आएगा ही नहीं सर अभी, इसलिए हम तक वो काम करेगा ही नहीं करेगा सर। इनकी अटकी है, हम लोगों की नहीं है सर। वही तो है सर, अब बोल दिया कोई बात नहीं, देखते हैं, बात की वैल्यू है सर हम लोगों की। और बढ़ाए जा रहे हैं सर ये लोग, ऐसा नहीं, इनके पास पता है 50 हो जाएंगे, ये लोग कर लेंगे। हाँ। नहीं करना कुछ नहीं है सर, अब हम लोगों को जो है वो है, हम लोग कुछ नहीं करेंगे अब।
Good evening, participants. My voice is audible. Please tell.
Yes, sir.
Okay, good. Uh, so we are going to start proteomics course today. Uh, I am Dr. Anand Kumar. I have completed my PhD from IIT BHU. Okay, so I am working as an assistant professor of biotechnology right now. Uh, so first of all, starting this course, you have to just give an introduction. Okay, in introduction, name, you belongs to which place, and you are doing which course, and in which stream. Okay, so you have to tell one by one. Okay, after that we will start this session. Clear? Is it clear to all?
Yes, sir.
Okay,
Yes, sir.
so please start. Dhriti Patel.
Hello, everyone. My name is Dhriti Patel. I am from Vadodara, Gujarat, and presently I have completed my bachelor's degree in biotechnology at Charuset University.
Okay, good. Next one.
Hello everyone, I am Priya and currently I am pursuing biochemistry honors graduate degree.
Okay, biochemistry honors. Okay, right.
Hello everyone. I'm Aisha. I'm from Hyderabad and I'm currently studying my BSc Biotechnology second year.
Okay, good. Next one. Please.
Good evening, everyone. I'm Niyati Saini. I'm from Pune, Maharashtra, and currently I'm pursuing my graduation course in biotechnology.
In graduation you are doing VTech or BSc?
No, I'm BSc third year.
Okay, good. Next.
Good evening, everybody. This is Falguni Srivastav, and I'm currently pursuing BTech in biotechnology, and in my second year. I'm also pursuing a summit research program along with my bachelor's from the same university.
Okay. You belongs to
I belong to Raipur.
Raipur. You belong to Raipur. Okay, good. Next.
Yeah.
Next one. Please. Next. Kajal. Kajal.
Good evening, sir.
Yes.
Good evening, sir. I'm pursuing my master's degree from NIPER, Hyderabad in pharmaceutical analysis.
Okay. So I think all have finished introduction. Okay. Next one is Ria Roy. Okay. Please.
So I am Ria Roy and I am pursuing biochemistry honors from New Delhi, India.
Biochemistry honors. Okay. From which institute?
Delhi University,
Okay.
Institute of
Okay.
Economics.
Delhi University. Okay, good. Next one. Kanimoji. Please introduce yourself. Please introduce yourself. Kanimoji.
Sir, I'm Kanimoli from Trichy, Tamil Nadu. I'm doing my master's in biotechnology first year in Holy Cross College, Trichy, Tamil Nadu.
Okay, okay. You are doing MTech or MSc?
MSc in biotechnology, sir.
Okay. Actually, how many of you are doing both courses like bioinformatics and proteomics? Please tell me if you are
I'm
participating.
doing it, sir.
Okay, one. And any other? Please tell. Only one, right? Are you participating in more than one course? Are you hearing my voice? Is it audible?
Yes, sir.
Okay. So you are participating in more than one course? Please tell me.
So no, sir, only one course.
Okay.
Sir, only one course.
Okay, okay. Actually, someone commenting that this link is for proteomics or bioinformatics. That's why I am asking. Someone have confused.
Sir, actually, you wrote in that Telegram group that this is for bioinformatics. So we got confused.
Okay. Actually, that was my mistake. Okay, thank you for confirming me. Okay, I got that thing. Please wait for a minute, okay? So we are going to start from basics, okay? So we will understand the basics today. it visible to all? Screen is visible? Please tell me.
Yes,
Yes,
sir.
sir.
Okay.
Yes, sir.
Okay, good. So first of all, we will understand about the proteomics. Okay. So before going to discuss about the proteomics, we have to understand the meaning of proteome. Okay. What proteome is. Okay. So the term proteome was coined in 1994 by an Australian postdoctoral fellow named Mark Wilkins. Okay, so proteome refers to the total set of proteins expressed in a given cell at a given time. Means uh, whatever the genes present in a cell, they are expressing a specific protein. So total proteins present in a cell, they are called as proteome. Okay, so here to study the dynamic protein products of the genome and their interaction. Okay, so a large scale characterization and functional analysis of the protein expression by a genome. So you know the central dogma. Okay, so in a central dogma we will study about the DNA to mRNA and mRNA to protein. Okay, so uh, if we are talking about the nucleic acid means DNA, so we will study about the genomics. Okay, if we are studying of DNA means genes, that we will study for the genomics in this branch. Okay, so if we are if we are studying of RNA, that that will be a branch of transcriptomics. If we are studying of proteins, that will be a branch of proteomics. Okay, so there are the branches. So proteomics, proteome is a defined as the complete set of proteins expressed during a cell's entire lifetime. Okay, so next, proteomics is the study of the proteome. It uses technology ranging from genetic analysis to mass spectrometry, right? Proteomics assesses activities, modifications, localization, and interactions of proteins in complexes. So next one you are seeing here the image. If you are studying DNA means chromosome, you are studying of genomics. Okay, you are studying of RNA means transcriptomics. If you are studying of proteins, this is a branch of proteomics. Okay, now we are going to discuss about the introduction part. So proteome indicates the total proteins expressed by a genome in a cell or tissue. Okay, now biomarkers detection might allow identification of patients who would benefit from further evaluation. Okay, with the development of proteomics techniques, proteome analysis provides a fast, non-invasive diagnostic tool for patients with various diseases. Okay. Next, the advent of highly sensitive proteomics technology can identify proteins associated with development of diseases well being before any clinically identifiable alterations. Okay. Next one, MS has a high resolving power and identifies proteins with more accuracy, right? Now, why we will study for, the proteomics? Okay, so genomics, role of genomics in a genomics, DNA tells what possibly. Okay, but in a transcriptomics we will study for the RNA. So RNA, what probably. Okay, but in a proteomics, what actually happens. So DNA to RNA formation by the process of transcription, and RNA to mRNA by processing, and mRNA to protein by the process of translation. Okay, after protein it will go for the proteolysis, post translational modification, and compartmentalization. Okay, so first, the behavior of gene products is difficult or impossible to predict from gene sequence. Second one, even if gene is transcribed, its expression may be regulated at the level of translation. Okay. Now, different types of proteomics are there. So we are discussing here about the structural proteomics, functional proteomics, expression proteomics. Okay. So in a structural proteomics, the ultimate aim of this proteomics is to build a body of structural information that will help predict the probable structure and potential function for almost any protein from knowledge of its coding sequence. Okay. So next one is the functional proteomics. It refers to the use of proteomics techniques to analyze the characteristics of molecular protein networks involved in a living cell. Okay. Next one is expression proteomics. It refers to the quantitative study of protein expression between sample differing by some variable. Okay. So next one is the proteomics objectives. Okay. So first one is the protein peptide separation. Next one is the identification and characterization of resolved proteins by MS. Next one is the data analysis and applications. Okay. So these are the objectives of proteomics. Now we are going to discuss about the structure of proteomics. There are four types of structure. Okay, primary, secondary, tertiary, and quaternary. You know, in a primary structure, there is a sequence of specific amino acid in a polypeptide chain. Okay, you are seeing here in a figure, these are the amino acid. Okay, like arginine, glycine, histidine, glutamine, glycine, alanine, arginine, aspartic acid, cysteine, leucine, isoleucine, tryptophan, proline, tyrosine, like that. Okay, so these are the amino acid chain. Okay, so we are calling it primary structure. Okay, now in the secondary structure, the primary polypeptide chain gets properly folded in the form of alpha helix, beta, plated sheet, random coils, and turns, right? So here there will be an interaction of hydrogen bonds. You are seeing here there is an interaction of hydrogen bonds, okay, in a secondary structure, and there will be alpha helix. If we will talk about the tertiary structure, then in a tertiary structure, secondary structure interacts with each other chemically to form a three-dimensional shape of the protein. Okay, so in a three-dimensional shape, alpha helix will be there and beta sheet will be there. You are seeing here, this is the helix. Okay, this is called as alpha helix. But in a beta sheet, these are the sheets. You are seeing here in a green color and blue color, alpha helix. Okay. In a quaternary structure, there is an interaction between different polypeptide unit. Okay, you are seeing here beta chain, alpha chain, Fe heme. Okay, so this is the quaternary structure. So you can see here experimental workflow, proper sample collection and storage, sample pre-processing, immunodepletion, protein concentration, etc. How you will prepare a sample. Okay, how you will analyze your protein through SDS-PAGE and 2D how you will fractionate your protein by LC, liquid chromatography, all these we will discuss. Now next one is the procedure of proteomics. Okay, so first is the separation of proteins, one dimensional electrophoresis, two dimensional electrophoresis, and multi-dimensional HPLC modern. Okay, after that we will go for the analysis of protein through mass spectrometry. This is a modern technique. Okay, after that database utilization. Okay, so now here there are 5 steps of proteome analysis. First is the sample collection, handling and storage. Second one is the separation of individual proteins by 2D electrophoresis. Next one is the protein characterization. Next one is the identification by mass spectrometry or other methods. Next one is the storage, manipulation and comparison of the data using bioinformatics, right? Next is the proteomics techniques. Okay, what type of techniques we will learn here in this course: gel based SDS-PAGE, 2D off gel based, uh, here LC, SCX, RPLC, reverse phase liquid chromatography, immunoaffinity. Next quantitative proteomics. Okay, please wait for a minute. Okay. You are seeing the same slide. My voice is audible.
Yes, sir.
You are seeing the proteomics techniques slide, right?
Sir, the slide is being shared.
Okay.
Yeah, sir. Now we can see.
Okay. Now it is visible, right? Proteomics techniques. So,
Yes, sir.
okay, here we will learn about the quantitative proteomics like iTrack, iCat, SELECT, MS, MALDI, LCMS, CEMS. Okay. Next, protein mixture from body fluid, tissue, food matter. Okay, we can go for the LC based workflow and gel based workflow. Okay, in LC based workflow, in solution digestion of protein mixture; in gel based workflow, protein separation on 1D or 2D gel. Okay, so in LC separation we will go for the peptides, and in gel digestion protein mixture. Okay, so after that we will go for the MS, analysis of peptides, ESI-MS instruments, multi-top. You know about this multi-top. Okay. After that, data analysis, protein identification, protein quantification, charged molecules, and beam are there. So now protein separation techniques, 1D and 2D. Okay, so what is SDS page? We will learn in a detail this thing. What is the purpose of doing gel electrophoresis? It has been seen that the running gel we are able to identify more proteins from the sample. An electric current is applied across the gel, causing proteins will differentially migrate based on their molecular mass. Okay, so you are seeing here isoelectric focusing gel means 2D. Okay, SDS page. So we will go in a detail here. So in isoelectric focusing, we prepare a gradient of pH, means low pH and high pH. You are seeing here at the bottom there is high pH and the top there is low pH. Okay. So next is the 2D gel electrophoresis. 2D gel electrophoresis is a method. 2D gel electrophoresis is a method for the separation and identification of proteins in a sample by displacement in 2 dimensions. Okay. First step is to separate based on charge or isoelectric point, called isoelectric focusing. Okay. Then separate based on size, SDS page. Okay. You are seeing here. Okay. Next thing is the separation of proteins by pi value, decreasing pi, soaking the gel in SDS solution and fitting it on a SDS PHL, separating the proteins by molecular mass with SDS page, decreasing pi, decreasing molecular mass, approximately 1000 E. coli proteins on a single 2D gel. Okay, after that we will learn about the HPLC, the basics of, you are seeing here basics of HPLC. Okay, so we will go in a detail. After that, mass spectrometry. Mass spectrometry is used for protein identification. It is useful to obtain structural information like peptide mass sequence. It is also useful in identifying type and location of protein modification. A mass spectrometer separates protein according to their mass to charge and by Z ratio. The molecule is first ionized. The process of ionization of proteins forces them to move towards the analyzer because of the change charges on ions. MS can provide molecular weight and structural information. MS always work with positive ions. Okay. Mass spectrometry. While 2D gel electrophoresis separates protein, it does not identify them. MS is used to identify them, which separates charged particles or ions according to mass. Okay. Two types of MS instruments, Malditov and ESI. Okay, will be used. You are seeing here image. Now you can see here components of mass spectrometer. Okay, there are three components. First is source, second is mass analyzer, third one is the detector. Okay, now after that we will go for the protein fingerprinting. Protein fingerprinting, also called peptide mass fingerprinting or peptide mapping. It is a technique for identification of proteins. Separated protein spots are obtained from the gel and then identified using protein fingerprinting. The method is based on the use of proteolytic enzyme to digest the protein into number of smaller peptide. Okay, unknown protein of interest cleaved into peptide by protease. Most commonly used protease is trypsin, which cuts protein at lysine and arginine position. You have to remember this thing. This is a very common point and you have to remember. When the digestion is incomplete, the set of peptides are produced. Okay, so peptide mass fingerprint. You are seeing here the image and process. And next one is the protein microarray. Another strategy for large-scale study of proteins similar to DNA microarray. Next procedure: a very small amount of different purified proteins are placed on a glass slide in a pattern of rows and columns. Followed addition of various types of probe molecules that are frozen dye-labeled to the array. When the probe binds to the label, it results in fluorescent signal that can be read by a laser scanner. Thus, this technique can detect thousands of protein interactions, can screen the ability of protein to bind other proteins in a complex receptors, antibodies, lipid, enzymes, hormones, specific DNA sequences, or small molecules such as new drugs. Right? So you can see here the image of microarray. Okay. So after that we will go for the protein-protein interaction like yeast to hybrid system. Okay. You can see here yeast to hybrid system for protein-protein interaction. Most of the yeast to hybrid system utilize the reconstitution of an active transcription factor to assay for protein-protein to make an interaction. The yeast to hybrid system uses the transcription process to make the prediction about protein interaction. The system requires that two yeast hybrids be prepared, called bad-pre system. The bad protein is fused to a transcription factor DNA binding domain. The other pre protein is fused to a transcription factor activation domain. Okay, when expressed in a yeast cell containing the appropriate reporter gene, interaction of the weight with the prey brings the DNA binding domain and the activation domain into close proximity, creating a functional transcription factor. The bad prey nomenclature has applied to in vitro method used to study protein analysis. In vitro method for protein interaction analysis are often employed to confirm interaction indicated by the yeast to hybrid method. Okay. You are seeing here the image of yeast to hybrid system. This is the process. You can understand basics. Okay, we will go in a detail in a later days. Okay. So after that we will go for the databases. Okay, data analysis search engines like Sequest, Mascot, Extendum, Peaks, and protein databases, and CBS, Swissprot. Okay, so in this course we will go for these topics. Okay, so this was the all about what we are going to discuss. Okay, so I think you got the basic idea of this course. After that we will go for the homology modeling like protein prediction in this course. So you have to be with a laptop or desktop because some softwares will not run in a mobile phone. Okay, so you have to arrange laptop, right? So we will end this session today here. So for the next round we will start from tomorrow. Okay, we will continue from tomorrow. Okay, if you have any doubt please ask.
Sir,
Yes.
sir, I
Yes, sir. I wanted to ask about the internship.
Okay, actually, I will discuss tomorrow about the internship. Okay, in an internship, you have to write a review paper. I'll tell you how to write review paper, what is the structure of review paper, and I'll suggest you some latest topic on which you have to write a review paper. Okay, and time to time, I will suggest you if you will face any difficulty in writing a review paper. Yeah. So about the
Okay, thank you, sir.